Okay, well, good morning. My name is Joe Gray, Joegray, and I'm Emeritus Professor of Biomedical Engineering at Oregon Health and Science University.
Dr. Gray, thanks so much for joining us today. I sure do appreciate it. Why don't you go ahead and we'll start and talk about OHSU and your role in the organization.
So I came to OHSU about 11 years ago now and founded what we then called the Center for Spatial Systems Biomedicine. It was a multidisciplinary enterprise that was aimed at trying to understand what the components of disease, especially cancer, are, the cells, the molecular components, how those components are organized in cancer, and how they change as cancers evolve. And the ultimate goal in all of this was to use that information to try to devise better treatment strategies for the long-term tolerable control of cancer.
So briefly describe your research projects involving microscopy and cancer.
Right. So one of the challenges in trying to understand cancer is that it's a very complex disease that involves changes in the organization of individual cells that make up the cancer, that changes or that involve the kinds of cells that make up the cancer, and it involves understanding how it is that those cells respond to cancer treatments. And so the goal of our research program was really to develop and deploy the analytical technologies that would be needed to understand what the components of the cells were, what the kinds of the cells were, how they're organized in tissues, and what constitutes a change in that tissue that would lead you in the direction of cancer control. And so the whole point of the program was to first deploy the measurement tools, and these are tools that allow us to measure the components, DNA sequencing, RNA sequencing, protein profiling, but more importantly, developing the imaging tools at the light and electron microscopy levels that allow us to really define the organizations of the components and the cells that comprise these tissues. And so first thing was to put those technologies into place. The second thing was to figure out how to manage the data, the massive amounts of data that these tools generate, and then finally to be able to interpret those data in ways that tell us about the mechanisms that are operative in the way these cancers behave and how it is that they respond to treatments. And the goal ultimately was to identify mechanisms of resistance and mechanisms of response, and to be able to figure out how to select treatment combinations really that will take advantage or exploit the vulnerabilities that the cancer cells have.
Describe the technologies and methodologies that you use to help to facilitate that research.
Right, so we operate in the center primarily at the light and electron microscopy level. So at the light microscopy level, we have deployed a suite of what we call multiplex immunostaining technologies that allow you to stain cells for a particular protein or protein combination, image them, erase the stain, pick a different protein, stain it again, image it, and so on, and to do this cyclically. And so after, a few tens of cycles, what you have is a multi-protein image of the cells that comprise the tissue. And so that is something that allows us to identify what the cells are that are in the tissues, and it tells us something about the molecular properties of those cells. At the same time, we've learned how to isolate the cells, isolate the components of the cells using nucleic acid and protein sequencing technologies, and to help us understand in greater depth what the molecular characteristics of the various cell types are. So that's sort of at the light and molecular levels. At the ultrastructure level, we have deployed a number of scanning electron microscopes that allow us to generate with resolutions down to about 4 nanometers what the various ultrastructural components of the cells are. What does the nucleus look like? What do the mitochondria look like? What do the structural proteins in the cell look like? In other words, what we're trying to do is to understand how cells are organized. And so the technologies that we use there, roughly speaking, are serial block-based imaging technologies where you use an electron microscope to scan the surface of a sample, get a high-resolution image, and then using one or another technologies to cut off a few nanometers of that surface, scan it again, cut off a few nanometers, scan it again, and so on. And this is more or less an automated process at this point, but it allows you to slice down through the tissue and generate what ultimately ends up as a three-dimensional model or image of that cell or that tissue. And that reveals some really remarkable facts about how it is that cells interact with each other and how it is that the cells, the components of the cells interact within the cell to exert cell function.
Well, at a high level, talk us through some of the key results that you've been able to deliver so far with your research at OHSU.
Well, I think probably the most interesting thing that we've done, or important thing that we've done really, is to take the suite of technologies that I just described to you and to deploy those clinically so that we can, for patients with metastatic cancers of various types, understand at all levels how it is that those cancers respond to treatment. And we have used that information to identify vulnerabilities that could then that would then direct us to select particular drugs for the treatment of that patient. And I think what's important here is that we do this rapidly enough that we can use the information to help the individual patient. So we've, it really does help us to understand not only what's going on in a cancer when we start treatment, but also what happens to that cancer as it evolves over time. And so that's given us, I think, a much better understanding of how it is that we lose control of a cancer and how it is that we can regain control of the cancer. This is the so-called SMART program that Gordon Mills is now directing. I initiated it, but SMART stands for Serial Measurements of Molecular and Architectural Responses to Treatment. And it really is a clinical program that allows us to follow what goes on in an individual patient over time as their cancer evolves and responds to treatment.
And what thesis are you looking forward to testing in the future at OHSU?
Of course, the challenge here is to identify what the operative mechanisms are in the cancers that cause it to become resistant to treatment and to figure out how to counter that. And I think that one of the really important things that our program has shown to date is that when one thinks about effectively treating a cancer, you have to understand what your treatments are doing to the cancer cells themselves, but also what they're doing to the non-cancer cells, the immune cells, the fibroblasts, the endothelial cells that make up the blood vessels, and so on. And I think that the challenge for the future is going to be able to figure out how it is that we can best kill the cancer cells directly, but also how we can manipulate the rest of the microenvironment so that it becomes anti-tumor. Reactivate the immune system, choke off the blood supply, et cetera, et cetera. And so I think the real challenge, that the program is looking forward to as we go forward is learning how to do both of those things effectively. And I think that we now have the measurement tools that teach us how to do that. What remains is to learn what the actual rules are that will systems biology, if you will, that's going to have to be learned in order for us to be able to do that effectively. That's the future.
And why is vibration and environmental noise a big challenge for your research at OHSU?
One of the opportunities with the technologies that we have is to be able to see with few nanometer resolution the features that make up these cells and tissues. And the challenge here, of course, is to make these measurements using technologies that run over periods of days to generate the data set. Well, if there's any vibration on the scale of a few nanometers, then it completely corrupts the images that we're generating. And so it's absolutely essential that the microscopes, especially the electron microscopes, be rock solid. And in Portland, this is a particular challenge because the building in which the microscopes are located is located right on the riverbank. And the soil is not rock, it's sort of squishy, and so it is prone to vibration. And so we've had to pay a lot of attention to literally building the foundation of the building and to installing vibration damping technologies to try to minimize that. And I think that we've been quite successful at that.
Good. And how has TMC vibration isolation helped to improve and ensure the standard research for OHSU?
Well, I think that one of the things, I talked about this as being part of a clinical program, but the technologies themselves are available throughout OHSU as core technologies. And so everybody on campus has an opportunity to use the tools and to generate high quality data. I think there are a number of groups that are particularly strongly engaged in this, and so they all benefit from having highly stable instruments. I mean, one of the instruments I haven't talked about, or types of instruments, are the cryo-electron microscopes that are part of the Pacific Northwest Cryo-Electron Microscopy Center. This is a national facility that takes projects in at even higher resolution, sort of angstrom level resolution, and determines protein structures as a service to the community. It's an NIH project. And so at that level, I think not only do the local users benefit from stability on these instruments, so does the national community.
Well, is there anything that you'd like to leave us with that we didn't touch base on?
Well, no, I think the message here that I'd like to leave everybody with is that we have finally gotten to the point in measurement science that we are able to define the components and the organizations of cells and tissues. And I think it's going to give us a lot of insight as to how the components work together to actually produce the functions that we observe in the cells and tissues that we look at in the light microscope. So it's an exciting time, lots to learn, a lot of challenges in terms of interpreting the data, but the measurement tools are there, and I think it's going to be fun to watch how the field evolves.
And very exciting. Dr. Gray, well, you've made tremendous contributions to cancer research, and it's not too much to say that you're on the leading edge. So I really want to thank you for your time today. It's been an exciting and an interesting interview.
I appreciate the opportunity. Thank you.
Thank you.